Hello and welcome to Chapter 10. This chapter we are learning about the origin and diversification of life on Earth. And if we're thinking about life on Earth, I guess it's really important to think about what it is that is alive. So let's take a look at just a few. This is just three of the 8 or 9 different things that are characteristics of life. Those are the ones I want you to know about. And they're important for this chapter. So the first characteristic is that all life is composed of cells. This means, by the way, that we don't define, at this time anyway, we don't define viruses as being alive. Even though they have a lot of the characteristics of life, we don't actually consider viruses to be alive. That's because they're not composed of cells. The next thing is that they have to have the ability to replicate. They have to be able to make copies of themselves. And finally, they have to carry out some sort of metabolism. And I know metabolism is a little bit of a squishy word, like we don't always know what it means. It means different things for different people. So when we say this with regard to cells, we're always talking about how cells carry out cellular processes like making proteins, making DNA, making things, breaking things down. That's what we mean by metabolism. So if these are three of the main characteristics of life, then we might ask ourselves, how did life evolve in the first place? How could you get life from no life if there's all these different characteristics that is life? So to start out with, we're going to take a quick look, a brief history of the planet, so you can kind of get a timeline here. And we'll be following this timeline a little bit more as we go along. So the first thing you should know is how old the Earth is. And we have this estimated to be about 4.5 billion years. You might hear 4.56 billion years, somewhere around in there. It's a long, long time ago. That's about when the solar system formed, and Earth formed at the same time as the rest of the solar system. The oldest rocks that we have here on Earth are about 3.8 billion years old. So there's a big stretch of time there where we're not sure what was going on. We've got some pretty good guesses, but if you don't have any rock, then it's really hard to have fossils. And so our oldest rock is only 3.8 billion years old. If you think about it this way, when the Earth first formed, it was molten, and molten does not equal solid rock. So it had to cool down and all this other good stuff. So then life, once there's rock and places substrate, if it's not molten, there's places for things to start to live. And so we do have the earliest life form in fossil, and that's about 3.4 to 3.5 billion years old. And this is a prokaryotic cell that's been fossilized. It looks a lot like a modern bacteria, but it lived about 3.5 billion years ago. This rock was found, these fossils were found in a rock in South Africa. Okay, so we got a little time frame there. We got some question marks about like what's going on in this time frame. So how did life start then? And we know certain things must be true. If the scientists are right, life must have evolved on Earth from non-living materials. And there's three really important things that had to happen in order for life to kind of emerge from non-living things. And the first thing is that there had to be formation of small molecules containing carbon and hydrogen. Carbon and hydrogen are the molecules or the atoms found in every molecule of living things. And carbon structure is really interesting because it can form a lot of highly variable molecules with lots of different functions. DNA, proteins, RNA, lipids, carbohydrates, all of these molecules are made up of carbon and hydrogen. So we've got to have small versions of these that can be put together. The second thing that we have to have is the formation of self-replicating information-containing molecules. And when we think about that, we tend to think about DNA. DNA is the information-containing molecule in all of our cells. And but we kind of think that maybe DNA wasn't the first self-replicating information-containing molecule. We think it was probably RNA, and I'll give you some more information on that later. Then finally, remember that all life is made up of cells, and cells are surrounded by membranes. So we have to have a development of a membrane. So all three of those things have to happen in order for life to evolve. And and it doesn't have to happen in any particular order. They just kind of all three have to come together. So what do we know? What evidence do we have that this is how it must have happened? And the first thing, of course, is you got to have these carbon and hydrogen rich molecules forming spontaneously. They have to form without living things being around, making them form. And it turns out that back in the 1950s, two scientists independently figured out the exact way that this could happen. Their names were Urey and Miller. And so I'm just going to describe what they did to figure this out. So they made this contraption, and again, they each kind of made the same kind of contraption independently of each other. They weren't really talking about it. And in this contraption, they put a few ingredients. And one ingredient they put in was water, and they heated that water up. So back in the 1950s, we thought that life must have started in these warm little ponds. Actually, that was something that came from Darwin. So, so we put in these warm water, we put in water and heated it up. Also threw in some hydrogen gas, which is H2, you can see right there, some methane, which is CH4, and some ammonia, NH3. And they put those in because back in the 1950s, we thought that those were some ingredients that might be found in the atmosphere in the early Earth. Notice that oxygen is not on this list. There was no oxygen in the atmosphere on the early Earth. Okay, so all this is heating up, and it goes up here, down through here, a little electrical charge, simulating lightning and so forth. Then it's cooled down, and it goes back in, the water goes back into a liquid form, cools down, and it goes over here, it's heated up, and it goes through this cycle. And they let that experiment run for not that long, within a matter of just a few days. So after a few days, they reached in with like a sterile probe and they pulled out some of the material that was in this cooled water. And what they found was amino acids. That's right, the building blocks of proteins have spontaneously formed with just a little heat, water, hydrogen gas, methane, and ammonia. That's kind of cool. And it was the first time that anybody had shown that organic molecules, those are the molecules made up of carbon and hydrogen, organic molecules can be forming spontaneously without living things making them. This is a huge deal. We now know that you can find these amino acids out on asteroids. You can find carbohydrates floating in space between the stars. I mean, life building blocks, the building blocks of life, they form spontaneously. And so it makes sense that life could form spontaneously. So, but that's just the first phase. We've got a few more things that have to happen. The next thing that has to happen is we need this self-replicating information-containing molecule. And as I said before, you've got to have a molecule that has this information like DNA, but the problem with DNA is that it can't replicate itself. I mean, it holds information, but it needs a huge number of enzymes to make copies of itself. And so we need a molecule that can self-replicate. And it turns out that there is a type of RNA that can do this. It's an enzyme. It can itself be an enzyme. And so if it can catalyze its own copying, then it fits this bill, and it turns out that exists. We have discovered this type of RNA. It's kind of cool. There's a great website called exploringorigins.org. If you're going to go to this website, make sure you're going to .org and not .com. So exploringorigins.org, and let's take a look at this website really quickly. It's got some really cool videos in it. I like a few of these, like this one's really cool. It shows how RNA molecules can form from RNA building blocks with just a little bit of clay.
Researchers have shown that clays, such as merlinite, are able to catalyze the formation of polymers of RNA from single nucleotides. Nucleotides, shown in white and blue, adsorb to the surface of a small clay particle, shown in brown. As the surface becomes more crowded, nucleotides that are close together can undergo a chemical reaction, resulting in the formation of a polymer, or strand, of RNA.
So this is something that has been observed in the lab. We see this kind of polymerization happening. And the building blocks of RNA are just those carbon and hydrogen-rich molecules. So if you want to explore this a little bit more, it talks more about how some of these can be enzymes themselves. All right, so that's the second phase. We get our self-replicating information-containing molecule, and we've already been able to see how they can form on clay and act as enzymes. The third step is to form a membrane, and I'm going to go back to that website showing you how membranes can form. Now, membranes are made up of fatty acids, and if you add a special molecule to that, you get your phospholipid. But let's take a look at how the fatty acids form. And just again, showing you what a fatty acid is compared to a phospholipid. They're very, very similar. And going down to the one that I like the best. There's a couple on here that I'm going to show you. This is how fatty acids can start to form a vesicle.
Under the proper conditions of concentration and pH, fatty acid micelles may join together to form a vesicle. Individual micelles first join to create a large, sheet-like bilayer. Random fluctuations of the fatty acid bilayer lead to the formation of a cup-like shape. When in close proximity, the ends of the sheet can seal together, forming a spherical vesicle.
So membranes can form spontaneously, is what this is saying, and we've been able to observe this in the lab. There's a couple of other videos here that you can look about how they can grow. Let's take a look at how a membrane can get bigger and bigger in this video.
When fatty acid micelles are added to a solution containing preformed vesicles, the vesicles are observed to grow rapidly. This is thought to happen through the formation of a micelle shell around the vesicle. Individual fatty acid molecules are then transferred from the micelle to the outer leaflet of the vesicle, resulting in vesicle growth and micelle shrinkage. Fatty acids must flip-flop between the inner and outer leaflet of the vesicle to ensure even growth of the vesicle membrane.
So the last of these is kind of putting it all together. And oops, sorry about that. Let's click on the link and show how you can get like these kind of cells that are kind of like protocells. They got a membrane, maybe they got some RNA on the inside, and what happens to make those replicate.
This animation shows the process of protocell growth and division. RNA nucleotides shown as blue dots traverse the protocell membrane and are used by an RNA replicase to make a copy of another RNA replicase. Meanwhile, the protocell membrane is constantly growing through the addition of micelles. Increasing the surface area faster than the volume causes the protocell shape to become elongated and unstable. The protocell eventually splits into daughter protocells, with the contents of the original protocell randomly divided between the daughters.
So the question is, are these protocells, as the video calls them, are they alive? And the answer to that is no. No, they're not alive. They're not really controlling their own metabolism yet, right? That's one of the prerequisites of life. And so, but it's so close. And these are the things that we've been able to see in the lab. If we can see these kinds of things in the lab, it's not hard to imagine how over the course of 1 billion years or so that this might happen on the Earth. And that's what we're going to look at now, is we're going to look at a timeline of the history of the Earth. So, like I said before, the Earth is 4.5-ish billion years old. So, and it was molten at that time. It's definitely cooled down after about a half a billion years or so. And at this time, about 4 billion years ago, we think that's when the Earth was dominated by these molecules, these self-replicating molecules like RNA, plenty of sugars and amino acids kind of hanging out, doing their thing. Um, Probably having protocells and actual cells by that time. But we don't have any record of it until 3.8 billion years ago, when the Earth cools, the first rock forms, and maybe this is when life arises. We don't have a fossil at this time. Hopefully, we'll discover new fossils, but we don't have a fossil until about 3.5 billion years ago. At this time, it takes about a billion years for something really important to happen, and that is for oxygen to start accumulating in the atmosphere. The only way that oxygen can accumulate in the atmosphere is if there's photosynthesis going on. And so photosynthesis started happening somewhere between 3.5 billion years ago and our known 2.5 billion years ago. This happened in prokaryotic cells like cyanobacteria. We don't see our first eukaryotic cell until about 2 billion years ago. And we don't see multicellular life until 1.5 billion years ago. So, and the life kind of chugs on for about a billion more years until about 600 million years ago. That's, an estimate. We have animals first evolving. There are literally no things that we would recognize as animals before 600 million years ago. And then in a short 100 million years, we get our first fish forming. Then plants coming onto land, followed by amphibians, insects, spiders coming onto land. We get our first reptiles at about 300 million years ago. And it doesn't take too much longer past that when we get our first dinosaurs. Mammals come shortly after the first dinosaurs, around 200 or 220 million years ago, depending on what estimates you look at. We have that followed by birds at 150 million years ago, flowering plants at 130 million years ago, and then at 65 million years ago, the dinosaurs die off. We get our first primates, which are a type of mammal, at 60 million years ago-ish. We get the first apes. Those are the ones without tails, at 20 million years ago. The genus Homo, that's the genus that we belong to, it was about 2.5 million years ago. And then Homo sapiens, that's the species that we belong to, evolved about 200,000 years ago. So just got a quick timeline for you. And I just mentioned that our species is called Homo sapiens, but really, we've been using that word species a lot. We've been throwing it around like we know what it means. And this is the chapter where we're going to talk about what a species really is. So what is a species? So there's a couple of sets of pictures here. There's these guys right here, and these right here, and these right here. And the question is, are these organisms the same species? And of course, we know like these two are the same species very clearly. They're humans, and if humans are good at anything, they're good at figuring out, what other humans are. We're not so good at looking at these two things that look very, very similar and figuring out that they're actually different species. And they are different species. We still have a good working definition of that. These two are also different species. Maybe a little bit more sense to you. This is a baby orangutan, and this is a golden lion tamarin. They look very similar, but not the same species. So there really isn't any characteristic that helps you decide whether something's the same species or not. We need to have some kind of good working definition. And so we do know that species in and of itself has the definition of different kinds of organisms. They're just different kinds of organisms. But like, how do you figure out if they're the same kind of different kinds of organism? And so actually biologists have several different ways of defining species, and that's why we have our definition of a species it's not just species. We got to have like this framework for it. So we're going to use the first definition. It's called the biological species concept. And all of our definitions of different ways of defining species are going to be called species concepts. So the biological species concept says that species are made up of populations of organisms that interbreed with each other. That means that we're defining species based on whether or not they can have sex with each other and have babies with each other. Um, we also say that maybe they could just possibly or potentially breed with each other, but it has to also be under natural conditions. And I'll show you an exception to this, something that's maybe not quite so natural in a minute. And we also have to explain with this concept that they have to be reproductively isolated, and that's a hugely important term for us, reproductively isolated from other such groups. We're going to spend a little bit of time talking about what that means to be reproductively isolated. All right, so well, that's a typo. That should say 3 key concepts. Actually or potentially interbreeding, natural populations, and reproductively isolated. And so I told you, it has to be natural, right? And this is a zorse, which is a hybrid between a zebra and a horse. And look, it's made this little baby zorse. It's awfully cute, but it's not really viable. It was bred in captivity, so it wasn't a natural population. And so that means that because it wasn't in the natural population, scientists would not consider the zorse to be evidence that zebras and horses are the same species. We would still call them separate species because in nature, zebras and horses are not breeding. And maybe you know why zebras and horses are not breeding in nature. Maybe this makes perfect sense to you if you know a little bit about geography. Because the truth is that zebras are found in Africa and horses are found in Asia, Europe, and now in North America. So they're not anywhere near each other in nature. And it turns out that that's really important. So what we have here is some sort of reproductive isolation by geography. And there's actually a whole list, a whole bunch of different kinds of reproductive barriers. You can categorize them as pre-zygotic, that literally means before a zygote forms. And they can be post-zygotic as well. That means after a zygote forms. So in other words, you're making a little baby there. So pre-zygotic barriers happen when individuals are either physically, literally unable to mate with each other, or if they are able to mate, the reproductive cells can't fertilize each other. And so they can't make a zygote, and so they are reproductively isolated in a pre-zygotic manner. However, sometimes you do get matings between things that we think are different species, and they produce these hybrid offspring. But if the hybrid offspring do not survive long after the fertilization process, then even though we do have a zygote forming, we still would consider them separate species because it's not viable. Alternatively, we might get the hybrid offspring surviving, and unfortunately, those hybrid offspring are infertile. They're or have reduced fertility. Like this mule here, mules are the offspring of horses and donkeys, and mules themselves are sterile, so they can't have babies themselves, which means that what that implies is that horses and donkeys are not the same species, right? They've got reproductive barriers. All right, so once we can figure out what our good definition is, and stand by because we've got some other definitions for you, we start naming species. And we rely on Carolus Linnaeus' system of naming species. He was the first one to come up with a system. We still rely on it a little bit, but we've elaborated since him back in, in the 1700s. We've kind of figured out a few more things. So, like, for example, Linnaeus didn't have this level domain that we have. And I've been talking to you about the domain level for a little while already this semester. The domain level is made up of three categories: domain bacteria, domain archaea, and domain eukarya. Domain eukarya is made up of any organism that has eukaryotic cells. And we're just going to kind of focus on the eukaryotes right now. Of the eukaryotes, there are four kingdoms. There's the protista. Those are the single-celled or simple multicellular things that kind of don't really fit into any of the other categories. There's the plants, the fungi, and the animals. And our friendly little zebra here is definitely an animal. Of the animals, there are approximately 30 some odd different phylum or phyla or phylums. And our lovely zebra here belongs to the phylum Chordata. In the phylum Chordata, there are those are the things with spinal cords. There are a lot of different organisms with spinal cords. You know, there's fish, and there's birds, and there's reptiles, and so forth. And there's mammals. Those are the ones with fur and feed their young milk. Of the mammals, and there's lots of different orders under the class Mammalia, we have our zebra belonging to the order Perissodactyla. And Perissodactyla refers to the shape of the foot here. It's got that hoof. And so that's Perissodactyla. Of course, there are lots of other orders of mammals, including the one that we belong to, which is the order Homininae. Next comes family, and this guy right here, he's in order Perissodactyla, but he belongs to the family Equidae. That's like all the equines. There's a genus, and that genus is oops, sorry about that. The genus is Equus. And then finally, we talk about the species. And the species is always made up of two parts. It's made up of the genus name and something called the specific epithet. The specific epithet refers to only a single species. So the second part is the specific epithet. The first part is the genus, and we call that the species name. And sorry about that. Keeps showing up there. Nothing I can do about that. But hopefully that makes sense to you. Yes, you need to memorize these terms. Luckily, I've got a mnemonic for you. And the mnemonic is: Dear King Philip came over for good spaghetti. Domain, kingdom, phylum, class, order, family, genus, specific epithet. And remember, the species name is made up of both the genus and the specific epithet. So if we were going to do this classification with humans, we would be, and like, let's just compare it to this this lovely zebra right here. We belong to the domain Eukarya, the kingdom Animalia, the phylum Chordata, the class Mammalia, the order Homininae, the family, oh my gosh, I've totally forgotten our family, the genus Homo, and the species is sapiens. So we're Homo sapiens. This just refers to some of the things I talked about. But I want to add that whenever you're writing down the scientific name of an organism like Homo sapiens or Equus quagga, you have to do it properly. And the first letter of the name is always capitalized in the genus. It's always capitalized. But the first letter of the specific epithet, that's always lowercase. And then you'll notice that this is italicized, and because it's kind of Latin and it's a foreign word, and you're supposed to italicize foreign words. And so you'll notice in lab in the next couple weeks that anytime you see a species name, it's made up of two parts: italicized or underlined if you're writing this in your notes, capital letter for the genus, lowercase letter for the species or specific epithet. All right, well, we're going to stop right here and get ready for part 2. See you in a few minutes.